Hey guys, hi. Welcome to Unacademy. This channel is Unacademy Future Doctors, and I'm your educator for biochemistry, Dr. Shivangi Shukla. Let me just check if the live stream has started. Just give me a minute to check if I'm visible and audible to everyone, right? Just give me a minute. Okay, so yeah. Is my video clear to everyone? Is my audio clearly audible? Is my voice clearly audible? Yeah. So I'm Dr. Shivangi Shukla. I'm your educator for biochemistry, and I'm here with another session tonight. That is clinical applications. This is a takeover from the last session that we were doing, which we had to break in between because the time was up. Okay, so this is regarding translation and fidelity of translation. Okay, that is one very important concept. Okay, so yeah, let us begin. I'll just quickly brief you about the different types of subscriptions for NEET PG that are available on the Unacademy app. Okay, the Plus and the Iconic subscription both are available on the Unacademy app. Both of these will give you access to video resources, video lectures from top educators across the country. Then live as well as recorded classes you can watch. And the USP is that you can watch these while on the go on any device of your choice, even at the comfort of your home, be it on your laptop, your tablet, your phone, or your PC, okay, whatever. The Iconic subscription will, in addition, give you access to resources from Prep Ladder as well. All right. Yeah, so Unacademy uses a lot of state-of-the-art tools in its special classes, plus classes, iconic classes. So that it can simulate, the video classes can simulate the physical classroom environment. Okay, and also it has come up with this rapid revision 2-month course, 2-month batch where you will have all. This is especially for all those who have to take the NEET PG examination this year. So between March 16th to May 16th, 2022, it is going to conduct a lot of tests, quizzes, and help you revise the entire 19 subjects in the field of medicine. Okay, for NEET PG 2022. All right, and while taking the subscription, do not forget to put in the educator code DRSHUKLA, Dr. Shukla. Okay, so this code is going to fetch you an additional 10% discount on both Plus as well as Iconic, on either of these. Okay, whenever you are planning to take the subscription, do not forget to put in the educator code DRSHUKLA while checking out. Okay, all right. So we had started with translation day before yesterday. Okay, now what is translation? How tRNA acts as the mediator or adapter molecule between the mRNA and the amino acids. All this we have already read. Now what we are going to read is, also we read about the genetic code, the characteristics of the genetic code, which are very, very important. Degeneracy, unambiguity, non-overlapping nature, non-punctuated nature, universality, and the exceptions to the rule of universality, that is the differences in the reading of the code between mitochondria and cytoplasm. Okay, then we read about post-transcriptional modifications, namely capping, tailing, and splicing. So all these important things we read. Now you have to start with the structure of tRNA. Okay, now what is tRNA, guys? tRNA is transfer RNA. Now what does it transfer? It is transfer RNA. Now why is this called transfer? Now again we have to focus on the nomenclature so that you never forget. A tRNA, as you knew, is the mediator molecule, the adapter molecule between mRNA and amino acids. Now, mRNA does not understand the language of amino acids. Amino acids do not understand the language of mRNA. So, they need to have someone who can associate with both. And that is tRNA. That is the adapter molecule tRNA. Okay. Now, what is tRNA? It is transfer RNA. What does it do? It transfers information. It transfers information between mRNA and amino acids. Okay. Between mRNA and amino acids. So this is what tRNA does. Okay. It is very aptly called the adapter molecule that will mediate the transfer of information between two different molecules that are not interrelated, like mRNA and amino acids. Okay, understood. Next, the first step in the adapter function of tRNA is charging of each tRNA molecule with its specific amino acid. Now, what is charging? Now, have you heard of those adapters that you have to place in whenever you change, like when you go abroad, you have to have certain adapters which will adjust your the incoming voltage to your to the requirement of your particular electronic gadget, which is fitted to work at a particular voltage that is supplied at our homes in our country. But in other countries, the voltage supply, the domestic voltage supply, the supply of electricity in houses is at a slightly lower or maybe slightly higher voltage. So the electronic device has to be adjusted. The electronic device is not made to function at that voltage. So what happens is the flow of electricity has to be regulated, and that is done by adapter. Okay. Similarly, here tRNA and its adapter function has to be charged first. How is it charged? Charged with an amino acid. Okay, you add an amino acid on the tRNA. Now, how do you add it? It is a very important process. Okay, you cannot just add any amino acid to the tRNA. What happens is there is a specific enzyme for each amino acid. Understood? For each amino acid, there is a specific enzyme. Okay. Now what happens is each 20 specific enzymes required for 20 amino acids will attach to specific tRNA molecules. Okay, so for methionine there is another enzyme, for valine there is another enzyme, for phenylalanine there is another enzyme. So there is a specific enzyme for, understood. So now let us see how it goes. Now this is the amino acid. Can you see this is the amino end and this is the acid end, carboxylic acid end. Now what will happen is, now this amino acid with its specific enzyme, which is amino acyl tRNA synthetase. Synthetase means it will require ATP. It will require ATP. I have always mentioned synthetase contrasted with synthase. Synthase does not require ATP. Synthetase requires ATP. So here what is happening is this particular enzyme, which is amino acyl tRNA synthetase, will then attach to this amino acid, and what will happen? It will make a complex with ATP because of course it requires ATP. And then this is like a covalent modification. Remember the chapter of enzymes which we read, where one of the one of the mechanisms of actions of enzymes was covalent modification. Similarly, here is what is happening. The enzyme, transiently the enzyme is getting associated with the intermediates, and then after the amino acid has been added onto the tRNA to form tRNA amino acid tRNA complex, tRNA amino acid complex, which is called amino acyl tRNA. The tRNA has now become amino acyl tRNA with the help of what? Amino acyl tRNA synthetase. How will you remember the name of this enzyme? This enzyme helps in the synthesis of amino acyl tRNA from tRNA. Okay, synthesis of amino-acyl tRNA from tRNA. What is the meaning of synthesis of amino-acyl tRNA? Addition of amino acid to tRNA will form amino-acyl tRNA. So the enzyme is very aptly called amino-acyl tRNA synthetase. Okay. Now this enzyme is going to add on to the amino acid. This enzyme is going to form a complex with the amino acid and ATP, and then go and so that the amino acid is charged. The amino acid has a is energized enough, and then that amino acid will be added on to tRNA to form amino-acyl tRNA, and then the enzyme and AMP will be removed and reutilized. Okay, so this is how. So what you have to remember is there's a unique amino-acyl tRNA synthetase for every amino acid. So for 20 amino acids, you have 20 different amino-acyl tRNA synthetases. Okay. Next, each specific tRNA differs from the others in its sequence of nucleotides. Got it? Now this tRNA is also what? It is RNA only. RNA is made up of nucleotides only. So each specific, now this tRNA also has specificity. Why? Because this tRNA has an anticodon region that binds to a particular mRNA, binds to a particular codon of an mRNA. Now how is it different? It is different because of the sequence of its nucleotides. Okay, so therefore tRNA is also unique. Amino acids are also unique. Amino-acyl tRNA synthetase is also unique. So there is so much of uniqueness, and that is what lends this process its accuracy. Okay, extensive folding of the primary structure of tRNA leads to the clover leaf secondary structure. What is the primary structure? Primary structure of protein is sequence of amino acids. And here the primary structure of tRNA is a straight chain. Okay, it's a normal chain. Okay, but this will extensively fold to produce a clover leaf secondary structure of tRNA. Okay, now this clover leaf structure, it is like this. Now this clover leaf structure, it has 4 arms. One is the acceptor arm. The acceptor arm is where the amino acid is attached to the this arm. The amino acid is attached to the acceptor arm, to the 3' CCA terminal. Okay, now every, like DNA, like mRNA, tRNA also has 5' and 3' end. Just like mRNA, just like DNA, tRNA also has a 5' end and a 3' end. Now, after extensive folding, you will see that one end is 5' and the other end is 3'. This is 5' say, and this one is 3'. So the 3' end has C, C and A, these nucleotides at the end. Okay, C, cytosine and adenine. Okay, so these 3 nucleotides at the end at the 3' terminal. And it is here where the amino acid goes and attaches. Then there is the D arm or the dihydrouracil arm. Its function you will read later on. Then there is the T-cis-C arm. Okay, that is T-cis-pseudouridine arm. Its function also you will read later on. And then there is the Anticodon arm, which, as I already told you, it will recognize the codon of the mRNA. Okay. Now see, this is the structure of tRNA. Clover leaf structure, as I told you. This is the dihydrouridine arm or D arm. This is the anticodon arm. This is the T-cis-C arm or pseudouridine arm. This is the extra arm, and this one is the acceptor arm. Now this is the 5' end. Go like this, and then this is the 3' end, which is the CCA terminal. And here the amino acid will attach. All of you should know how to draw the clover leaf structure of tRNA. It is extremely important, guys. Okay, the clover leaf structure of tRNA is very, very important. Okay, next. So this is the anticodon arm. Now the mRNA is read from the 5' to the 3' direction. Okay, but the anticodon arm, because it is opposite, it is complementary to the codon. Anticodon arm is complementary to the codon sequence on the anticodon arm contains nucleotides that are complementary to the triplet codon present on the mRNA. Okay. Now, the anticodon region contains 7 nucleotides, out of which these 3 are the anticodon regions that are complementary to the triplet codon on the mRNA. Now, this codon you read from the 5' to the 3' direction, and the anticodon you read from the 3' to the 5' direction. See, this one is the 3' direction like this. You read from the 3' to the 5' direction. So, please remember how to draw this. If you draw it like this T size C arm will come here, D arm will come here. Now this has become upside down. Always remember how to draw the structure of tRNA. It is extremely important, guys, that you know how to draw the structure. Because once you see it, you feel that yes, you know, you understand. But once you start drawing it, then you know that there are few intricacies that you seem to have overlooked. Okay, so 5', 3', where different arms are situated. Okay, those are the things that you should focus on. As I already told you, the anticodon region contains 7 nucleotides. Okay, now this region is what recognizes the 3-letter codon on the mRNA. The mRNA is read in the 5' to 3' direction, and the anticodon region is read in the 3' to 5' direction. Okay. The sequence in the anticodon region is read from the 3' to 5' direction in the anticodon loop. Now, what are these 7 nucleotides? They are, the first one is a variable base. The second one is a modified purine. What are purines? Pure silver, pure Ag, purine, modified purine. Purine means pure silver. This is the mnemonic. So, A and G are purines. So, either it can be A or G. Then there is XYZ. This is the anticodon region that is complementary to the codon, triplet codon that is present on the mRNA. Then there are 2 pyrimidines. Pyrimidines are T and C or U. Okay. Variable based, modified purine, XYZ, pyrimidine. Okay. Now, codon and anticodon are antiparallel. We already know this. Codon is read from the 5' to 3' direction. Anticodon is read from the 3' to 5' direction. Okay. Next, ribothymidine pseudouridine cytidine arm. That is T-Cy-C arm. Ribothymidine pseudouridine cytidine arm. T-Cy-C arm. It is involved in the binding of amino acyl tRNA. What is amino acyl tRNA? It is the charged tRNA, okay, the tRNA that was charged with the amino acid, with the unique amino acid, particular amino acid, using the enzyme amino acyl tRNA synthetase, okay. So, this T-Cy-C arm is involved in binding of amino acyl tRNA to the ribosomal surface at the site of protein synthesis. Now, what did I tell you? This tRNA is the adapter molecule, okay. Now, this tRNA that is charged with the amino acid and it has bound to the, it is reading the mRNA. It has to attach to the ribosomal surface. Okay, I will see later on. Okay, because ribosomes are what? Ribosomes are the site of protein synthesis. So, for protein synthesis, this tRNA will have to attach to ribosomes, and that is done through this T-Cy-C arm. Okay, you'll know this. You will understand once we go ahead. Now, this D arm is the recognition of a given. D arm is dihydrouracil arm. It is there for recognition of a given tRNA species by its specific amino acyl tRNA synthetase. Now you know this. Now you understand how a particular enzyme will recognize its particular, how here in charging, during charging, this particular amino acyl tRNA synthetase will recognize its particular unique tRNA. How will it do that? By the D arm, dihydrouracil arm. Okay. Next is the acceptor arm, which is the site of attachment of the specific amino acid to the 3' end. And the 3' end contains what? CCA. Understood? Now comes the Wobble hypothesis. What is the Wobble hypothesis? We already read what is degeneracy. Now, because the genetic code is a triplet, Therefore, we have a lot of codons. We have extra codons to code for amino acids, isn't it? 61 codons for 20 amino acids, barring the 3 stop codons. So what happens is more than one codon is available for one amino acid. So one amino acid can have more than one codon. This is what is called degeneracy. Okay, now there is something very special about degeneracy, that is the degeneracy of the genetic code resides mostly in the last nucleotide of the triplet. Now what does this mean? What happens is, suppose one amino acid is coded for by ACU and another and this amino acid also is coded for AAU. No, sorry. See, suppose one amino acid is coded for by ACA. Okay, now this can also have ACU and ACC. So suppose these are the three triplet codons that code for one amino acid, that is X. Okay, now there are more than one codons, only one amino acid. So this is what? This is degeneracy. But there is something special about degeneracy, and that is the degeneracy is in the third place. The degeneracy is in the last nucleotide only. And you see, the rest of them are same. The difference is in the last nucleotide. So this is what is the Wobble hypothesis. What happens is, you have a particular tRNA, unique tRNA. You have one unique amino acid. Okay, but the codon on the mRNA can be different. Okay, so if you so the anticodon, now the corresponding anticodon for ACA will be what? Come on, the corresponding anticodon for ACA will be UGU. For this one will be UGA. For this one will be UGG. Okay, so while the amino acid is X, there is one unique amino acid. So the amino acyl tRNA synthetase will also be unique. That will be the same. And the tRNA will also be unique. Okay, so what will happen is that the tRNA is the same. The tRNA suppose has the anticodon region as UGU. Okay, so if the MR, now this one is the tRNA. Okay, this one is the tRNA. This is the anticodon region with UGU. It has recognized this mRNA with ACA. And this has been charged with a particular enzyme, amino acyl tRNA synthetase, that is Y. Okay, and this is carrying this X amino acid. Understood? The amino acid is unique. Y enzyme is unique. This tRNA is unique because it carries UGU anticodon to ACA. But now suppose there is another sequence on the mRNA, that is ACU. This ACU is also for X only. It is coding for this only. So the same enzyme system will come in because the amino acid is the same. So the enzyme will be the same, tRNA will be the same because they are unique for that amino acid. Now this tRNA had the anticodon region UGU. So now this base pairing rule, the Watson-Crick base pairing rule, has been violated at the third nucleotide. Understood? Here it is the same, but because of degeneracy at the third place, what has happened is the Watson-Crick base pairing rule has been violated, and this is what is the Wobble hypothesis. Okay? Therefore, pairing of the codon and the anticodon can wobble at this specific site, at the third nucleotide of the codon. Each tRNA molecule contains a specific anticodon to a codon on the mRNA. For a given codon in the mRNA, only a single species of tRNA molecule possesses the proper anticodon. For the other degenerate triplet codons, this particular tRNA will only come in. So it is complementary to only one of these three. For the rest of the two, the Watson-Crick base pairing rules will be violated because there is only one unique tRNA, one unique enzyme, amino acyl tRNA synthetase for this X amino acid. Understood? Okay, but some tRNA can utilize the anticodon to recognize more than one codon. Okay, this is what we are reading. Alright, understood or not? Wobble hypothesis is very, very important. Now, let us take the example here. Okay, the example is arginine. Okay, now arginine can be coded for by AGA also. This is on the mRNA, and by AGG also on the mRNA. Now, what is the anticodon region on the particular unique tRNA? Now, arginine is X here. Y is the enzyme, that is amino acyl tRNA synthetase, that is going to charge the unique tRNA for arginine, that is X. Now X is unique, this tRNA, the unique tRNA can be Z. So X, Y and Z, both of them are unique. Okay. Now what happens is, this is the mRNA containing AGA. Okay. And this one is the tRNA containing this tRNA is Z. And this amino acid here, arginine, on the 3' end. Sorry, this is the 3' end right here. Arginine on the 3' end. You remember, this is the 5' end of mRNA, this is 3' end. This is the anticodon region, or the tRNA is read from the 3' end to the 5' end. Okay, this is the CCA terminal carrying the amino acid arginine, which is X, which is unique. Now for arginine, every time any code for arginine is read on mRNA, this tRNA will be called in, utilizing the charging by this enzyme. This enzyme will first charge this tRNA with X, and then this tRNA will be brought in. Now this tRNA has UCU on the anticodon region. UCU can very easily recognize AGA according to complementary Watson-Crick base pairing rule. But now suppose AGG has come in here. Now AGG also the body will interpret as, the cell will interpret as arginine only. Now arginine is X, it is unique. So just after it recognizes it as arginine, it will recruit this unique tRNA Z. This unique tRNA Z containing this anticodon region UCU has been brought in. It has been charged prior by this Y only, all the unique systems. Now it will see that this is U, okay, U and A, G and C, very good. But now you see G and U have to pair together. U and G have to pair together. Okay, so this is what is degeneracy. This is what is wobbling of at the last nucleotide. Anubhav, hi, what is the part that you wish to read again, understand again? See, I'll explain the Wobble hypothesis again. See, what happens is there is degeneracy of the genetic code. That means more than one triplet codons can code for one amino acid. Okay, if the example is, you can consider arginine. Arginine can be coded for by AGA also and AGG also. Both of these will mean arginine only in the body, okay, in the cell. But as we have read, tRNA is unique. Okay, each amino acid has only one tRNA and one amino acyl tRNA synthetase. This enzyme is used to charge the particular tRNA with a particular amino acid. So this is arginine. Arginine say X. So X has one particular tRNA to itself. Okay, no other tRNA can be given for the synthesis of X. So just one tRNA is allotted for arginine, and that tRNA is Z. Okay, this is an example. And this Z will be associated with X utilizing a unique enzyme, that is Y. So this XYZ system is unique. Okay, it cannot be changed. Even if this arginine has made new friends, they will not accommodate, they will not change Y and Z. Okay, they will remain the same. So this arginine, now suppose what happens is the mRNA is read from the 5' to 3' direction. So it is AGA. And the tRNA is read from the 3' to 5' direction. So suppose this is the genetic code AGA. As soon as the cell encounters this genetic code, as soon as it reads it, it will understand that this is arginine. Then it will ask for its complementary, its unique tRNA. What was the unique tRNA for arginine? It was this Z. So this Z tRNA is brought in. Now this Z tRNA is charged by Y, and Y will add what? The unique amino acid, that is X. So this Z tRNA has already been added with X, that is arginine. It has been charged with arginine. Now it will come and it will read this AGA and it will pair with it. This already has UCU. So it is A is complementary to U, G is complementary to C, A is complementary to U. Very good. So Watson-Crick base pairing rules are fulfilled here. They are abided by here. But now suppose further on reading the mRNA, the cell encounters AGG. Now again the cell will say AGG is arginine only, AGG is X only. So the friends of X, that are Y and Z, they are again recruited. Now they are the same. Okay, they are the same. No matter whether X is called AGA or X is called AGG. The friends will remain the same. Z and Y. Z has UCU on the anticodon region only. So now when it is going to pair with it, U is complementary to A, C is complementary to G, but U is not complementary to G, but it has to pair with it. So this is what is violation of Watson-Crick base pairing rule at the third base position, third nucleotide position, and this is what is called the Wobble hypothesis. Understood this? This is because of degeneracy of the genetic code. Okay, all right. So now we have to begin with translation proper. So we'll start it in the next class because the time is up for this one. Okay, I hope you guys have understood. This is a very important concept, Wobble hypothesis. All right, I hope there is no doubt now. Anubhav, have you understood this? Then we can continue from the proper steps of translation in the next class, and then you'll be able to understand this one even better once you know how translation proceeds. Okay, understood? If there's any doubt, you can ask me. Otherwise, tomorrow I have similarly I have 3 sessions tomorrow as well on YouTube, and 2 sessions I'll have on the Unacademy app from tomorrow. Okay, so a lot of sessions, exhaustive studying, because exams are near. So let us all go full throttle and study. Okay, and all right. So if there's any doubt, you can ask me. Also, do not forget to subscribe to this channel, Unacademy Future Doctors, and do not forget to follow me on the Unacademy app. Go to the Unacademy app, NEET PG section. There, look for Shivangi Shukla, and you can follow me there for timely updates of all special classes on Unacademy. Okay, guys. Then the floor is open for any discussion, any sort of doubts that you have. You can ask me in the next 1 minute. Otherwise, let's call it a day then, if there's no doubt. All right. there's any doubt, you can ask me. Alright then guys, see you tomorrow in the back-to-back YouTube sessions and the Unacademy special classes. Okay, bye-bye. Thank you. Take care. Good night. See you.